Well, thanks for the invitation, Graham. As I said when you asked me to come and talk, I really only feel qualified to talk about one of these subjects because TB and SPR training is a difficult juggling act and I dropped one of the balls on the way. But I'll tell you about what research I did, and it's been highly entertaining, and I advise you not to go down this route if you want to make a good salary and become a consultant. So why do TB research? Well, there are lots of reasons, but one of them is that you don't end up like this cartoon character, lying awake at night, asking why am I here? And the reason for that is because you're working with a major killer pathogen for which we've all got a lot of respect. And if you're working with a pig, streptococcus, or with dandruff-causing agents, then sometimes it's harder to get out of bed in the morning than it is when you're working on MTB. So it's a good bet. Second good reason to do TB research is that TB is not going away. It's not going away in London, as you know. This graph finished in 2003, but you know the trajectory of that line of TB notifications in London continues to rise. And it's not going away globally either, and it's unlikely to go away during any of our research careers unless we have a really brilliant idea. And even if we have that, then it took, remember, 300 years to eradicate smallpox. So TB's not going anywhere, and you can get into it and probably stay in it for a whole research career. A third recreational reason to go into TB research is that TB happens in lots of fun parts of the world. Newham, which is where I spent most of my TB career research life, but also various other exotic locations, some of which here are coloured darker blue than others. But even if it's a pale blue, you can find little pockets of dark blue within that pale blue to entertain yourself. So here are a few of the destinations where I've worked. This is the Welcome Centre, but only one L. That's in Newham, where I did most of the work on my PhD. This is Europe's largest prison, 5,000 bed, or 5,000 inmates, I should say, remand prison in St. Petersburg. I tried to set up a trial of vitamin D supplementation to prevent TB. This is at Yurt in Mongolia, where I enjoyed a lot of fermented yak's milk, while also trying to set up a study in the prison there. This is Cape Town, where I finished off my PhD working with Rob Wilkinson in the lab there. And this is India. Hardly need to say that, but where the next project is just taken off. So I'll tell you a little bit about those now. So just to illustrate this personal view, I'll tell you what I did. So I finished my house jobs and did a DTM&H in Liverpool, and then decided to go and do some SHO jobs for a couple of years in South Africa. So I did a casualty job in Soweto and then worked for a year out in rural South Africa doing a bit of everything. And that's when I really became interested in TB. There was a whole TB ward there. We didn't have culture facilities, just smear and chest radiography and clinical acumen in larger or smaller quantities. But certainly it was an exciting time to be working in TB, and I came back to the UK thinking, well, this is something that would be really interesting to research on. So I did my MRCP back in Liverpool, and then saw an advert in the BMJ for a teaching job in Newham, teaching the medical year of the dental course to dental students in East London, which took up 2.5 days a week, and the other 2.5 days one could then spend on spare time setting up a research project. And so I started reading around, trying to find a research question to answer. And I came across this paper from Rob Wilkinson and colleagues in the Lancet 2000, which was a pretty simple case control study, actually, done in Norfolk Park. Rob Davidson, who was the senior author on the paper, measured vitamin D concentration in a cohort of TB patients, all of Gujarati ethnic origin, with active disease, and an ethically matched cohort of controls who were all tuberculin positive but didn't develop active TB, and showed that profound deficiency was significantly more common among the patients than the controls. So far so good, but there are several potential explanations of this phenomenon. It could be that the egg is vitamin D deficiency, and that leads to reactivation of latent infection and active disease. Or vice versa, it could be that active TB somehow
causes vitamin D deficiency, for example, by making you go out less into the sunshine, making you eat less, and perhaps even, as some of our lab work is now showing, the pathogen may induce vitamin D deficiency as a means of subverting the immune response. So to try and tease out this chicken and egg situation, a clinical trial was needed. And this was the trial that I got the train down to Euston to do. We're going to get a large number of people with latent TB infection, randomise them to vitamin D or placebo, follow them up for 3 years and compare rates of active disease between the two arms. Simple enough, you might think, until you meet a medical statistician. And they'll tell you that you need to randomise over 13,000 patients to demonstrate a 20% difference between groups. And this is a function of the fact that even in recently exposed young people, you won't see a much higher rate than 5% over 2 years of active TB. So what you're trying to do is demonstrate the effects of an intervention between 5% versus 4%, and to show such a small absolute size difference, even though it's large proportionally, you need a huge number of patients. So that was when I called up Robert and said, look, I want to do this trial. Have you got any ideas about what we could do? And we sat down and decided to keep the front end of this trial, but instead of looking for the development of active TB as the dependent, as the outcome measure, we compared the immune response using a whole blood assay. So randomised 200 TB contacts to get vitamin D or placebo, take their blood, incubate it with mycobacteria ex vivo, and measure the ability of that blood to contain mycobacterial growth. And the results of that in a nutshell are that people who received vitamin D 6 weeks prior to this blood test were slightly better able to contain bacterial growth than those who received placebo 6 weeks previously. And if they're vitamin D deficient to start with, then that difference was greater. Now, this stuff isn't rifampicin, don't get me wrong, I'm not making any claims that it's a wonder drug, but there is obviously some evidence of signal here, which at a public health level, when you have a large population with a high prevalence of vitamin D deficiency, if one supplements, then one might see some sort of public health benefit. So I spent 3 years on that project, and then on the back of that and some lab work we did showing mechanisms by which vitamin D enhances macrophage ability to contain mycobacterial infection, we got a small grant from the British Lung Foundation to do a second clinical trial, this time using adjunctive therapy of vitamin D in patients with pulmonary tuberculosis. And this study I did with Graham and other collaborators around London. And I tried to then go back and having finished my research and not written up my PhD, I then decided to try and go back into clinical medicine. And this was a complete disaster because I was trying to do general medicine on call in Harlow while my research nurse was struggling to recruit patients to this clinical trial. And then the MHRA, who's the drug regulatory authority in the UK, decided to inspect my trial, and basically the wheels came off. So I had to decide research or clinical, and I went for research. So I went back and rescued the trial, and the MHRA ended up saying some polite things about it, which perhaps they wouldn't have been moved to if I had still been on call in Harlow. But it didn't mean I didn't have a salary, so as researchers do when they don't have salaries, they write a grant. And we then, together with Chris Griffiths and colleagues in the Centre for Primary Care, wrote a programme grant to trials of vitamin D in patients with asthma and COPD. This was because there was emerging evidence that the antimicrobial effects of vitamin D weren't just restricted to TB, but also it could prevent upper respiratory infections, which of course are the major precipitants of asthma and COPD exacerbations. So we were lucky enough to get the cash for that, and that then kept me in business for another 5 years, in which time I actually did manage to write up, much to my supervisor's delight. And then in 2013, we submitted a grant to the MRC to do that original trial that I wanted to do 10 years ago, a trial of vitamin D for TB prevention. And this is now being funded, and it's called the VidiKids trial. It'll be taken off in India later in the year. We're randomising 7,000 school children to get vitamin D or placebo, with the primary outcome of acquisition of latent TB infection. So here are my tips on research. You can take them or leave them. But I think J. Paul Getty really has got it. You've got to work hard, but you can't guarantee striking oil. You've got to try and optimise your chances, obviously, by reading as many papers as you can to find the best research question. But I'll be the first to admit that there's an element of luck in this, and your success will depend on whether or not, to a small extent, it will depend on whether or not you get positive results from your early studies. So there is an element of luck here. In terms of, sort of early on, I think reading some good journals is a good idea, and not necessarily just about TB, but just read the Lancet on the tube on the way to work and back, and you get an idea of what high-impact journals are looking for and what are the hot questions that are out there at the moment. It certainly helps to get funding for a fellowship if you've got a couple of papers. They don't need to be randomised trials. You can write case reports, reviews, systematic reviews, something like that. But if a funding body can see that you can put pen to paper and then push it through the editorial process, they'll be much more inclined to waft some money in your direction for a research training fellowship. I think finding a good supervisor is really important. It's got to be somebody who suits you. You want to go, there's a whole spectrum of supervision, from somebody who's completely on your back looking at your results the whole time, to somebody who's a lot more relaxed and just lets you get on with it but isn't necessarily available. You've got to decide which phenotype of supervisor you like and pick him or her. What I would say, though, is that you're going to be spending a lot of your life on this project, so if you can have a major hand in developing it, then that's all to the good. And in particular, if you're writing a research training fellowship, really, if you can come up with the basic idea yourself and write a lot of that fellowship yourself, then you'll end up essentially writing your own job description, which is a pretty nice position to be in life. I think it's also worth checking out what the situation is regarding funding, how much of your salary is going to be funded, how much money is available for consumables and that sort of thing, because it's quite easy to run out of cash, especially if your plans are grandiose. In terms of once you've embarked on your project, this is, I think, is the biggie for me, trying to get the right balance between clinical and research duties. It's a tricky one. I found that actually, it wasn't until I actually gave up clinical altogether that I could really spend time with my macrophages and really get into the in vitro aspects, which eventually yielded some decent publications. But I'm not very good multitasker, so it might be that you are better at juggling the two aspects of life. But I think when you go in, you need to have some sort of plan about how you're going to keep your clinical duties going, because this is where I fell down. If your study can't be big, make it detailed. I think it is often difficult to do very big studies, especially when you're starting out, because they tend to be very expensive. But you can measure a lot. And this sort of ties in with my second, my third point here: own the statistical analysis of your data. I think it's really worth learning some, not just basic stats, how to do a couple of t-tests or Mann-Whitney tests, but to really sort of get to grips with multivariable statistical analysis, so that you can generate your data and then really waterboard it and get the most that you can out of it and get some biological insights from your data. If you pass it off to a medical statistician, they're often too busy to look at it for the next 6 months, and they won't love and care for it in the same way that you will. Another thing, make space to write up. It came as a bit of a surprise to me that people were so bothered about whether or not I wrote up my PhD. Now that I've got a few PhD students, I've realised why they were bothered. It's because if you've got PhD students who don't write up, then you get penalised by the research funding bodies and by your own university. So for your supervisor's sake, at least, write up. But also, if you have jettisoned your clinical career, then you've got only your academic career to rely on. You need to be able to say you've got a PhD so you can progress. But I was a bit surprised to find out that was important. The final thing, I think this applies all the way through: hone your research question. Make it something you really care about. And really, a good research question should be something that you can put in a sentence. So I've put up a few questions that I'm interested in at the moment. Why are some TB contacts apparently resistant to acquiring latent infection? I'll put that one up for Graham. We can talk about it in the pub later, Graham. Why do interferon gamma release assays revert from positive to negative in some TB contacts, and what's the clinical significance of this phenomenon? What role do neutrophils play in the immune response to TB infection? What's the role of adjunctive vitamin D in the treatment of multi-drug resistant TB? And is baldness an evolutionary adaptation to optimise vitamin D status in older males? So if any of these questions interest you, and you fancy doing some research with me, that's my email address. Cheers.